amazed and blessed by his presentation this evening. What we'll do is we'll give him some time, 30, 45 minutes, to just talk to you about the cell events, and then we'll take a little break. We're going to take our love offering as soon as he is finished. Let you run to the bathroom, let him run to the bathroom, and then we'll come back and do a Q&A session immediately after that. So everybody's good to go for the question and answer. So that's our procedure for this evening. So at this point, let's pray together, and I'll turn it over to Dr. Rathman. Father, we thank you for the opportunity of being in your house, of listening, Lord, to one of your servants whom you've given skill and ability to talk about the evidence for you, Lord, that's found even in the smallest components of life. And so I pray that you would bless Dr. Rathman this evening. Speak to us, Lord, who are here assembled, that we might understand your will and your workings. Thank you, Lord, for servants like Brother Terry, who's come to share with us this evening. In Jesus' name, amen. All right, Terry, God bless you.
It's indeed an honor and a privilege to have some guinea pigs to talk to. Talking to the mirror does not work. I can do it in 15 minutes. I can do it in an hour and a half. We're going to find out how long it takes right now. And don't be afraid. I'll be hanging here as long as you want to stay afterwards to answer your questions. Hopefully you will have more questions, because the more I study this, the more questions I have. And that's the way it goes. Same thing in the scriptures. The more you dig, the more treasures there are. You can already see I changed the title, right? Right? Now, to some of you, it's going to be Greek. That's what this is. To some of you, it's going to be Greek. But I'm trying to make a point. As mentioned this morning, you're going to see chemistry of life that involves translation. So if you're talking about translation, you're talking about a mind being involved here. You're talking about words that have meaning. It's already a mind, but we're talking about translation. Something molecular doing translation. Also, I guess you noticed, we're calling this the Studies in General Revelation. Everybody know what that is? That's the other book that you've all talked about. That's what's generally available to everybody. That's what everybody's going to be responsible to, answer to when they stand before the Maker. It says, Romans 1:20, you are without a excuse. No excuses. None. Anyway, this is Greek. So, with the help of Translate Google, I translated for you. The chemistry of life. I changed the title a bit. I took the word complexity out. Why? Because I want you to be the judge. Does it look really complex? All right? Let you decide. That's your judgment. Don't let us make an assertion for you that there is complexity. Now we're talking molecular complexity. I'd much rather be talking about the chemistry that was in my life, talking lithium chemistry. That's much easier. This is far more difficult. But in my life, you already recognize that. That's somebody doing a reaction in glassware. Notice stirrer, all kinds of setup. Well, I don't want to lose you. In organic chemistry, they have recipes. Recipes. Thousands of them. And what happened with, I like this one because somebody publishes a recipe, they get somebody else to check it. And what's really interesting, when they do these checks, they find out, you know, even if we get the same molecule, but from a different producer, it doesn't work. So what I'm already telling you, you've got to have pure stuff, at least pure enough to do what you're trying to do. Now, this is a keepsake. This is my grandmother's, Pennsylvania Dutch. Pennsylvania Dutch. She was the best baker for miles, all right? And if you think about it, if you can think baking, hey, that's chemistry, believe it or not. And it's the most exacting type of food preparation. You can't make a mistake. Both of these have apparatus. See all the apparati? Both of them have apparatus, right? See how similar they are? We're drawing analogies. This is the second one. We're talking language analogy. We're talking baking and organic synthesis. They both obviously have ingredients and the amounts. What's so amazing about this, she has some of my favorite recipes in here. She doesn't even include the amounts. And that's so critical, right? And, well, then there's also the medium. That could be water. But some of my favorite cookies are, you know, the favorite. Have you made cookies this past Christmas? My favorite is when I'm making cookies, butter. Put the sugar on, beat it up real good. Oh, throw some oatmeal in, okay, I'll make it healthy now. But you have to have a medium, right? A medium in which to do this reaction. Same thing as organic synthesis. Instructions. My grandmother has some cookies in here called the chocolate macaroons. They were incredible. They looked like there was an earthquake. Of course, there was a pecan in the middle of it, and it was glossy. I like cookies that crunch. Don't you all like them like crunch? But as soon as you started chewing, it turned to fudge. She has the ingredients for that chocolate macaroon, but no recipe, how to put them all together. And I tried. I don't think when I took care of Mom, that's when I found this. I said, Mom, we gotta make these. We tried 20 different ways, never got it. Now we figured out how to make cookies hard, cookies soft, all that good stuff. But baking, that's science. Something as simple as science. Now also, obviously you gotta have temperature, order of addition, how you add them, that's critical. By the way, DNA does all that for you, okay? You have to also have the workup. Now, in organic synthesis, many times the yields are very low. We were talking earlier. When I go visit a place to consult, we have this reaction. We want to sell this product, but we're only getting 35% yield. Well, that's not very good. Guess what the yield was for the best science there was for making amino acids from what they thought was primordial earth. The best yield was 2%. What's the other 98%? Believe it or not, that 98% is injurious to the 2% you just made. Now, baking, that has good yield. You take the cake out, right? Plop. It's about 99% yield. So baking has pretty good yield. Final thing before we go to the next slide. For both those recipes, in order for those to happen, what do you need? You need a chemist, right? You need a baker. You need grandma. Get in there, grandma, do this. Guess what? With the chemistry of life, you don't even need that. It supplies that. It supplies what people call enzymes, proteins, same difference, okay? And in fact, they call them workhorses. You got that? Workhorses. We have in our cells, we have enzymes doing these things. They are the bakers and the organic chemists, already supplied. So, does the complexity of the chemistry of life infer intelligent design? Maybe I could stop now, but you say no, it doesn't, okay? It doesn't. But in this discussion, what comes up immediately? Immediately what comes up, well, especially for the other paradigm, the evolutionists, now wait a minute. You can't just say there's an intelligent design, you need proof. Well, one person that started this debate, oh, I skipped something here. Intelligent design, why not call it intelligent causation? Think of the word intelligent design. I probably shouldn't even get into this. Intelligent design, that seems redundant. Something that is intelligent, that is a design, and something that's designed is intelligent. So I think if science is all about cause and effect, it should be intelligent causation or intelligent origination. Just a thought, for whatever it's worth. Or let's get right to the point. Why not just call it intelligent designer, right? Or intelligent agency. You know, in the Cosmo game, they always talk about there is some kind of intelligent agency, some kind of an agency out there, a transcending agency doing it. You notice if you have intelligent agency, you could call it the intelligent agency of creation, the CIA. No. Well, anyway, Back in the 1700s, there was a man named William Paley, a theologian. And this guy was into the general revelation, the second book, the other book, that which is generally available. And he studied nature. And he brought all that into his theology. And so one day he said, well, gee, if I'm walking along and I find this watch, I pick it up, right away, suppose he never even saw it before, wow, somebody must have made this, right? It didn't just happen. It didn't just happen. And so he would say, well, gee, this is just like all the stuff I've been studying in nature. It all works together. That must be like a clock itself. So there's an analogy. So he put together a formal argument. This is not like I had an argument with my wife, all right? He put a syllogism together, premises and a conclusion. And that's how this goes. He says objects in nature are analogous to man-made machines. Man-made machines are the result of intelligent causation. Analogous effects will have analogous causes. Cause and effect, that's science. Therefore, objects in nature are the result of something analogous to intelligent causation. Sounds pretty good to me, but wait a minute. We're not in a boxing ring, but it almost is. But back then, they were just having these discussions, very civil. Actually, prior to William Paley, there was a gentleman named David Hume. And he said, anytime you're trying to draw an analogy, the things which you're trying to draw an analogy with, they must have similar attributes. And the more similar attributes, the better the analogous. And he said, wait a minute, you can't compare a clock with a living organism. With a cell, you can't do that. All right? I'll go along with that. Sure, they're different. So, what happens today, we have a guy named Richard Dawkins. This is about the 3rd edition of the Blind Watchmaker. He's responding to that. He says, yeah, I'll go along with the watchmaker, but he's blind, which means it's just by chance and natural selection, whatever that means. Natural selection is causing the watch to be created. He's written, of course, like I just showed you, The Blind Watchmaker, and you can tell you from this book he's written, The God Delusion, what paradigm he's from. He's probably the leading atheist in the world. He is a professor of biology at Cambridge. I don't know if you ever know, but there's another gentleman, John Lennox, he's on the left there. And just like, he's Irish, but just like two Englishmen, if you might let me do that, they have had several debates together. And it sounds like two guys talking with a cup of tea. And it's so great to listen to these guys. You should listen to these. You should actually read, if you can, read The Blind Watchmaker. I can give you a PDF where you can get it online for nothing, if you want to read it, all right? He is one very good writer. He even wrote a book on how to write. But John is also a good writer, and he's kind of retaliating. These are two good books I read during Christmas. That was my Christmas present, read two books. And one was called God's Undertaker, and it has Science Buried God. Excellent book. I'm just going to point out these two, and Gunning for God. So, we're talking about this watch. Is the watch really a good analogy for what we know about the cell today? Let's take a look. But before we do that, out of that book, here is an interesting statement. Chapter 5, out of the Blind Watchmaker, to explain the origin of DNA protein machine by invoking a supernatural designer is to explain precisely nothing, for it leaves unexplained the origin of the designer. To me, that is saying, I'm worried about this. He's worried about this, because he's already come up with an excuse. Well, if it really looks like we think, like there is a designer there, how do we know? That is a real designer. Maybe there's somebody else. Maybe there's another designer. The problem with most atheists, and it's our fault, he doesn't have the right concept of God. He says we believe in the God of the gaps. No, ladies and gentlemen, We believe in the God of the whole thing. And that's what people need to understand. He doesn't understand that. They make a straw man God, and they think they're shooting him down. What we're going to focus on is the DNA protein machine, as promised. This is what does the translating. Remember, we're talking translating. This is the only thing we can focus on tonight. And hopefully by tomorrow morning when we finish this, you'll know. Or my body will be hanging out there on, you don't have an oak tree out there, do you? The DNA protein machine. Actually, it's a DNA protein transcription, translation, language machine. All right? So that's our focus. And just looking at that, I'm going to say, does that look complex enough? Now, you can get this, you can see this online. Roche puts it out. There are two charts, part 2, part 1. This one talks about the molecular processes. I doubt if you can read that, but you're going to see the words again. In the middle there is your translation. All right? And I relate to calorie bags. There's also something, if you're going to translate something from, the cell does something first, a transcription. Now don't worry about it, we're going to see this more often. These are part of the DNA protein machine, a translation and transcription. We're not going to talk about replication. Certainly that's important. Proteins and DNA are involved in that. This is a quick close-in look. And if, I want you not to be able to see it too well. Fortunately, it's out of focus. You can't see it well. But there are all kinds of reaction arrows going every different way. And it's tying each of those brown blocks together. It looks almost like a schematic of some sort for a computer. So we'll totally block it out. This is the one that you need to kind of get in your mind and take home in your head. And actually, it was Watson and Crick, actually, Francis Crick, he came up with what he called the central dogma back in 1958, all right? This is what he showed in sort of a cartoon arrangement here. Now we're going to, instead of calling it the central dogma of biochemistry, we're going to stick with our title called The Dogma of Chemistry of Life, all right? Plus, we're not going to even talk about replication, even though that was included in his central dogma. And the reason I want to include it, did you ever take a watch home that you found, and the next morning you look in the dresser, there's 2 of them, 3 of them, 4 of them? That's what this is talking about. So I think we need to stay away from that, because I think David Hume's right. Maybe we can't compare a cell to a watch, you know, or watches. So we're going to look at going from DNA, right there, and our goal is to make protein, or proteins, all right? DNA to proteins, that's our goal. How's it done? First, you use DNA, and I like to call that the hidden language of life. This is the Greek, you might say, inside the cell, okay, in the nucleus. That's in the nucleus, the hidden language. And what's going to happen is, we're going to transcribe, see the word transcription? We're going to convert the DNA to RNA. And we're going to make a working copy of half of the DNA. You notice I have a little apostrophe there? RNA is like DNA, but RNA is like a dialect of our Greek. It looks like it, but it doesn't have all the same letters in that language. So that's why I like to call it, it's half of the DNA, and it also looks like DNA, but it isn't. It has slightly different letters, a slight modification. So we're going from the hidden language to a working copy, RNA, and it's done by transcribing. And what it's doing it is RNA polymerase. Now, anytime you see, I'm sure you're going to be reading biochemical journals tomorrow, anytime you see an ASE, that means it's an enzyme. Let me say something quick about enzymes. Enzymes are proteins. Now, probably when you think about proteins, you're thinking about hamburgers. That is protein, but that's a different form. It's formed in a different way, all right? We're not even having a chance to talk about hamburgers tonight, all right? This is a different way to move the amino acids that make up protein, And we're going to learn more about the structure of it. And this RNA polymerase is the workhorse. If you remember anything, there's workhorses, right? So the next thing we do, the cell does, it takes the working copy, takes the RNA, and it does the translation. There's no more Greek, shall we say. It's going to put it into English. And I say that's the observed language. That's what we see, all right? That's what we see around us. And it can be proteinaceous, meaning meat or that kind of, but it also means enzymes. Now you notice this doesn't have the ASE on it. It's slightly different. It has enzymes or protein in it, but it also has, believe it or not, and I have here a horse, but it also has an oxidant. It has a different type of thing. It has RNA, a different kind of RNA in it to make the ribosome. And we're going to see a movie at the end of this that will summarize all this to show this incredible step of translation. And we're going to talk a little bit more about what the ribosome looks like. I would be cheating you if we didn't talk about chart 1. And I just have it you can't see it very well. This is one part, I call it the power dogma. This is where energy is made. It takes energy to run this. It actually makes energy to run it, but it gets a little bit of energy left over from doing that. And if you can see closely, I don't know if you can make it out, see those arrows? Those are actually arrows pointing in either direction. This whole thing can move backwards, forwards, and there's all kinds of other reactions tied to it. This is the energy. Sometimes it's called the Krebs cycle. Obviously, it's a cycle. It's also called the citrate cycle. And it makes ATP, which is the hay. Over every one of these arrows, and actually if you looked at, when you go online, you can use the icon on the left there to go look closer and closer and closer. There are actually some places there are 4 or 5 arrows there. Every arrow indicates that there's a protein or an enzyme involved. Every one of them. So now we're not just talking a couple of horses or animals that working animals. We're talking many animals involved. All right. To get a real good understanding and appreciation of the protein DNA machine, we have to go to take the BCC 104 intermolecular non-binding refresher. And I got approval from this. We can do this. Got to explain a little bit. Everybody knows molecules, right? These are little amino acid molecules. The white, the yellow, the red are all atoms. The red is oxygen. Blue is nitrogen. The yellow is sulfur. The white is hydrogen. All right? And of course, black is carbon. That's the real driver. Carbon. We're all carbon. What's that? Carbon footprint? All right? Now, what holds these all together is what? What holds all these atoms together? Electrons. Electrons are molecular orbitals for the molecule, and they are orbiting throughout the molecule. And as they orbit, they have specific places to go. It holds the atoms together, hence a molecule. There are electrons buzzing around you all right now. When I clap my hands, I'm clapping electron clouds together. If you do it enough, it gets hot, because they don't like it. Well, at least I got applauded one time already tonight. Okay. All right. So, we have molecular bonds that hold atoms together, but we're going to talk about interactions between molecules. They're just as important. Obviously, it's important to hold a molecule together because everything we're talking about are molecules, and even huge molecules, right? But it's just as important to learn about the intermolecular non-bonding. They're not bonding things together. Believe it or not, things can interact. That's good. So we're going to have a little bit of this background because I know you're thinking, where's he going in this? Where's my DNA? Believe me, it'll pay off in the end with hard work. Everything does. Now, here is a major law. I'm hoping you got this. That's why this is BCC 104. Major law in physics concerning plus and negative charges. Who knows what happens if you have a negative and positive charge? What happens? Oh, they attract. This is a smart crowd. And of course, just looking at the way I put the sentences together, like charges repel. You guys know as much as I know. I might as well give up. That's fantastic. But on a molecular level, a little bit more of a view now. We all know that electrons are what charge? Negative, correct? And the electrons, we already talked about, they go around the nucleus, which is positive, right? Electrons orbit the atomic nucleus, or when in a molecule, the entire molecule. Nuclei of atoms are positively charged. The nucleus inside there, right? Everybody knows that. That's positive charge. Are you positive?
Yes.
All right, good. We don't want anybody to be negative around here. Oh, that's an electron. Do we have an electron here? No. All right, but there are a lot of free electrons around. Some call them free radicals. So, another thing, if you have a neutral molecule, you have then an equal number of positive and negative charges, right? Makes sense? They cancel each other out. It's a neutral molecule. We talked a little bit about this morning. So you guys are ready to go. You think we're going to go to the land of DNA? Sorry, we're not. We're going to water. Simple water. Yeah, here's my water molecule. You can see, this is the, I can't hold it. This is oxygen. Oxygen is usually red on the models, all right? And here are your hydrogens. So H2, two of them, and oxygen. Now if you look at it, there's also something else here. And on the screen there, it shows that, oh, there's a pretty strong location of negative charge, right? And that's where most of the density on this part of the oxygen molecule are pointing, all right? And notice also, back to Greek, that word means partial. It's only a partial negative charge. It's not a full negative charge, but it's just enough. I wish I knew the number, but I don't know it. But the thing about it is, we have a partial negative charge on this side because electrons are negative, all right? And we have a partial negative charge on this side of the oxygen because those are negative. Now, same thing down the bottom. What's happening in the electrons are being pulled more towards the oxygen, and it leaves a little positive charge at the end of each hydrogen. So, if I have two negative charges, where is the average negative charge? It's somewhere in the middle, right? If I have two negative charges, if I wanted an average, it would be somewhere in the middle. If I have two of these down here, positive charge, where would they be? They would be right here. What the physicists tell us, or chemists, they'll tell us that's a permanent dipole, and that results from the arrangement of the atoms and the arrangements of the electrons. Are you all with me? You got a dipole. And because it's a dipole, I also like to say it's a permanent net dipole, because you have to average all these vectors of plus to minus when the electron is moving. It turns out, on the average, that's where the dipole is. Water is the most unique molecule in my mind. At least atom molecule, for the amount of atoms I have, it's the most phenomenal. Do a little study some afternoon, some week on water. But anyway, we have a dipole. Now, this is a space-filling model of water, H, two H's, and one O. Now, I can't show it, but right where the M is on the word, can you see the M? That's where a good bit of the electron density is. It doesn't show that. And on the other side is some more. So I'm still kind of showing the arrangement of the densities of negative charges and positive charges. There's positive charge down here and a positive charge down here. All right? Now, what happens, I have here some water. All right? And what happens in solution, those dipoles are interacting, but because of thermal energy, they keep moving. But occasionally, they line up perfectly. You know, plus to negative, negative plus. The dipoles are adding up together. All right? So what happens now, this is a certain volume of the water molecules. Now, stay with me, please. The water molecules, all right? Now, as things slow down, we don't see the dipole-dipole interaction. We see more of what is called hydrogen bonding in water. This is very important in understanding DNA, and that's why I'm beating myself to death on this, and probably beating y'all. But anyway, what happens, at a certain point in time, remember I said there's a little partial positive right here? Well, there's also a partial negative here. They line up, oxygen, hydrogen, and you have a partial positive, and then over here, you have the negative. They line up because of the charges. You may say, well, why doesn't it get any closer? Well, because there's only a small partial positive charge there. All right? And the rest is electron density. So it's being attracted but still pulled, pushed away at the same time. So it's held. They don't go against each other. Does that make sense? There's a positive charge, it attracts it there, but it can't get any closer because there's a lot more electron density, electron cloud there. So it only gets a certain length, and it's a constant, consistent length. Now, once you freeze, if you look down at the bottom of ice, what happens when it freezes, all the water lines up, all the hydrogen bonds line up. All of them line up just like that in a straight line. You have oxygen, hydrogen, the interaction space, and then you have that electron density, a lot of times shown by a single line. Now, to give you an idea of how small that positive site is on the hydrogen, all right? I wish I had a computer program to do the calculation and with the wave functions and show you how it's really shaped. I need a drink anyway. This was all figured out ahead of time. I'm not going to explode. I just drank 5 times 10 to the 24 water molecules. That's 5 with 24 zeros going that way. Now, if it was frozen, if I ate an ice cube, they would be all lined up. See, I'm trying to give you a feel for how small that spot is. And yet it's very critical when you start talking about DNA. Because, I have one slide here. Can you read that? This is not a miracle. Look at me. The dipole-dipole interactions and hydrogen bonding on water molecules are holding me up. All right? The interactions, the dipole-dipole interactions of the water molecules, this would be the, let's see here. This would be a dipole-dipole interaction of the water molecules. And this would be a hydrogen bonding. And because at the surface, the molecules can concentrate with their charges horizontally and downward, it ends up being what some people like to call a skin. This is the reason you can skim a rock across the water, because they're all concentrated at the top in terms of lining up. And that's how comes that bug can stand on it. Now, okay, in the last slide, we said hydrogen bonding for water molecules goes like this. O, and there's a hydrogen bond, and hydrogen, all right? Then we have the dots that show the interaction, and then the electron density on the oxygen. Well, you have tons and tons, billions of these interactions in DNA. So not only can you have oxygen, hydrogen, and oxygen, you notice this is nitrogen, right? Everybody knows that's nitrogen, right? This is nitrogen. You can have a nitrogen, hydrogen, oxygen arrangement. And notice here you can have this. And what's amazing, that spacing is exact. Exact. And what happens in DNA, you're going to see more about this. You have two different types of compounds. They call them bases. Sometimes they're mistakenly called nucleotides. But they have one called guanine and cytosine. So remember, all you got to remember is G and C stay together. You can think in your own mind. I have a way to remember that in my mind. I think of gas chromatography. If you watch my cousin Lenny, remember it was GCMS, mass spectrometry, that put the crooks in, freed his cousin. Anyway, so we have hydrogen bonding. Some places we have 3 hydrogen bonds between 2 of these bases, and some places we only have 2. Which you think is the stronger? The one with 3, right? Exactly. All right. Now, again, guanine, cytosine, G and C, A and T in DNA, they go together. I remember that by the telephone company, AT&T, right? I remember that. Now, that's for DNA. For RNA, you have the letters. Actually, they're characters. You have the letters U, A, G, and C. And if you reverse it, you can remember that U is always substituted for T because UT is right around here. It's all around here. I see that abbreviation everywhere. You have University of, what is it, Tennessee? Yeah, right? So you always, from now on, you know, well, I know how RNA and DNA differ. DNA has GC, AT. RNA has bases that are GC, A, U, right? You with me? Okay, that's RNA. Okay, we're only getting, there's other non-bonding intermolecular forces, interactions that you can have. We already talked about water, and if see another molecule, this is methanol. See, it has oxygen, hydrogen, all right? And they're both polar molecules because they have dipole moments, all right? And in this one, they're showing hydrogen bonding going on. Now, this interaction between methanol and water is saying that methanol is hydrophilic. Do you know what that means? Water, hydro, water, philic, loves, right? Philic. So it's hydrophilic. So a lot of times you'll see people say, well, try this stuff. It's very hydrophilic. It means it loves water. It'll dissolve in water, right? Now, we don't want to look at all these. We just want to look at one other one that's really important, and that's down here. We have polar molecules up here. We have non-polar molecules here. This is octane and hexane. It's stuff you find in gasoline. Does water mix in your gasoline? Very little bit. That's because of some of these other forces. Very little bit does. So what happens is non-polar molecules also have an interaction, which is so amazing. And what they are, they're instantaneous dipoles. And what is happening, the electrons are moving, these two molecules approach each other. The one electron cloud pushes the electron cloud away from the other molecule and causes an instantaneous positive charge. So what's happening is a collision of two molecules. They're causing one another to be attracted because of moving and forming instantaneous dipoles. Now, there's a word you got to take a look. This is called dispersion forces. There's another word you got. I'm sure. Does everybody know what the word hydrophobic? That means it doesn't like water. That means if this doesn't like water, you think these two guys like each other? Things which are hydrophobic like each other. All right? If I had a last name, never mind. Hydrophobic. That means they don't like water, but they like themselves. Two molecules which are nonpolar, but by their interactions, they can create an instantaneous polarity, and they're attracted to each other. Now, I know I beat you to death on this, but every one of you has millions and billions and trillions of membranes in your body. And what holds those together is this interaction. And if you look closely, it has, this is a phospholipid bilayer, and these are long strands of these type molecules. I have another slide coming up to show a little bit better. But you see those like dark yellow? Those are chains coming down from the bilayer phospholipid. Now these things, you see it says polar group. Now if something is polar, what molecule that you know now might be there? What molecule? Water, right? It's a polar end. So because water is polar, so this is a hydrophilic side of water, of the membrane. Now, there's also another molecule in there. You see that there? Looks like a honeycomb. This is also found in membranes. And it's bilayer. You see one set and it's turned upside down and they're attracted to each other. That's a very hydrophobic region in between the polars, the red circles, all right? And I'm going to tell you a little bit more about the long chains. Again, these are just like what we saw for the octane and the hexane, hydrogen molecules, and they're being attracted in by these instantaneous dipoles. Now there are some of these long chains that actually, this one has a kink in it. But first I'm going to say one thing. The last slide showed about intermolecular hydrophobic attractions, all right? But this one, this is intramolecular dispersion forces because these are all part of the same molecule. Like interstate, that connects the state together, right? The roads do. Well here, now we have an intrastate. Maybe you've had a state roads, those are intrastates. Now if you notice, some of these long chains have a kink in it. They have a bad D, Pastor, and it's permanent. Now why would the designer do that? That's a bad design. Come on. Stay with me. If you remember the little honeycomb thing I pointed out? Now if you could just keep your eyes to the left. What that is, believe it or not, ladies and gentlemen, is cholesterol. You need cholesterol in all your membranes. Did you know that? How many knew that? Oh, you're smart. You need that. Well, if you look at it, this is a very rigid region of this cholesterol molecule. Imagine 4 lawn chairs, those beach kind, and you've got duct tape and you started connecting them together, it would be a rigid mess. And that's what this is like. It's like 4 chairs all stuck together with duct tape. Very rigid, all right? But then, you see, this is also like very flexible. This is just like a hydrocarbon chain. And hydrocarbon means it's just a molecule that consists, in this case it's a moiety of the cholesterol. It just contains carbon and hydrogen, just like gasoline. So we have a rigid part. Oh, look at this. OH. What does that tell you already? It's a polar end. Where do you think that's going to go? Is it going to point towards the polar end of the phospholipid? Yes. And what else could be there? Water. So, now this is the reason for going through all this. We have these phospholipids with the long chains, and they're doing this moving. But then, every so often, you have the cholesterol molecule in there. That gives rigidity to the membrane, and it's precisely needed. You can't just have phospholipids for a membrane in life. And by the way, scientists have not figured out where these came from. Not figured out at all, if you didn't have life to begin with. So the cholesterol fits in there very nicely. And this is a little bit more detailed to the membrane. All the blue circles, those are the polar ends where water might be. And if you look in there, you can see the cholesterol, those are the yellow. And the other ones are the phospholipids with those chains. And so this is a very hydrophobic area, right? Very hydrophobic. Doesn't like water. You notice here's a protein going through it, and it has a hydrophobic segment. It says that. It fits right in there perfectly. These are like addresses on a cell. The body needs to send something somewhere. Another place in the body says, okay, put the address on, send it to this cell. It's communication between cells. There's a lot going on around here. All right. Everybody stand up. What time is it anyway? Is that what time it is? Whoa. You've been so good. All right. Put your hands up. All right. That's good. Put them out in front. Bring them down. Bend your elbows. Bring your hands in like this. That is chirality. Staring you in the face. Right there. You may sit down. You did well. You know what chirality is. All right? I'm going to have to speed up here. I cannot believe the time has flown by like this. So let me just... Let me just go right to the amino acids. There's chirality all around us. Our body, turns out, only has one amino acid. Nature, they have shown by experiments that you can make amino acids, but you always get a mixture of two. Scientists have yet to figure out how can we only get the L amino acid, all right? Here are some of the amino acids. I should have, if you notice, go back. We have, on the amino acid, the amino is this amine group, the acid group is this. And there is an R group, and here's a hydrogen. I was going to spend some time showing you that why those two are different. These two are chiral. What that means is there are mirror images. If I put one of these in a mirror, it would look just like this. There are mirror images, and they're non-superimposable. I cannot make them, if I try and do that way, you can see those are two out of the road. They're non-super, they're completely different arranged configuration of atoms in space. And that's why it's hard to do it. The pharmaceutical industry spends billions of dollars each year trying to make the left or the right for pharmaceuticals. Billions of dollars. I slipped right through one here. Nature makes chiral molecules real simple. That one has 17 chiral centers. Here's all your amino acids. Now, amino acids just aren't used for proteins, enzymes, workhorses. Like, for instance, quickly, this is tyrosine. If I put an OH here and an OH here in the right chiral space, I have something I don't really need right now. Maybe you do. I have adrenaline. So what I'm saying, amino acids are also used for other things in your body, not just for building enzymes and proteins. Okay, well, you can't read it that well anyway. So what happens is, you take your amino acids and you form polymers. You react one amino acid after another. The body does that and it lines them up to make chains. The chains actually have a spiral. As you can see here, has a spiral, or they call it the alpha helix. And what actually happens too also, after you get so long, this unit fits right in here. It goes to a specific shape, all right, a specific shape. And what even more astounding is, you can have one of these and 4 of them fit together. go here. Here's a better picture of the alpha helix. It's turning like this, and I skipped over it because I'm hurrying now. If you look at this chain of amino acids, what happens is you see that oxygen and the electrons, and you see this hydrogen and nitrogen. What can happen is you get hydrogen bonding, right? And that's what happens in the alpha helix. You have hydrogen bonding in the alpha helix of this protein. And you can't see it that well, unfortunately, but you have that attraction. That helps hold it together. And here are the R groups extended out from the alpha helix. This is an example of hemoglobin. It's made up of four proteins, consists of 574 amino acids. It's four units, 2 different types put together, like I said. And what do you think holds those 4 units together? What holds them together is hydrogen bonding and dispersion forces, the hydrophobic forces. So the forces we talked about of hydrogen bonding and dispersion forces hold that together. It even holds the heme part. This is the part of the hemoglobin that holds the oxygen. Quickly, Richard Dawkins, although I think Richard Carrier on infidels.org, he says, he's quoting Richard Dawkins as saying, well, that's not a big deal. That's only 574, but I think only 190 are essential. 190 essential, and that's a lot to be at the right spot. Chiral sugars. In DNA, we have ribose. Now, if you look at it, if you see here, on this carbon number 2, is an OH. That's also called an alcohol. All right? And here's carbon 3, carbon 4, carbon 5. There are actually, in this particular structure, there are 1, 2, 3, 4 chiral centers. There are 16 possibilities to have those atoms arranged in that way. And only the body uses this particular one. By the way, nobody knows where these came from. Only cells make these. Only cells make it. Now that's the sugar that goes into RNA. Here's the sugar that goes into DNA. All right? And you notice the difference. The big difference is this. There is no oxygen. You see the oxygen here? There's no oxygen here. It was deoxied. It was taken out of there. That's how comes they call it deoxyribose. It's the same thing as ribose, but it's lost that oxygen. I want to go on. This is a DNA molecule. Since we're behind here, I want to just show you. I mentioned this morning, and I'm just going to probably stop here. The DNA molecule, like 5 to 6 feet long, if you could stretch it out. Here we have the double helix of the DNA, and what happens is, you even have an electron microscope of it. This is 2 nanometers, just multiply by 10 to get in angstroms. That's angstrom. You measure molecules in angstrom units. And here the protein is a protein that the DNA wraps around. Another, and these are called histones, all right? And the DNA wraps around them. Once it's wrapped around them, make these nucleosomes, they then stack them. Then once they're stacked, they start making loops. And that's put onto another protein scaffold. Now what you have to think and keep in mind here is, where is the information to make that protein? It's in the DNA, right? It's in the DNA. So where did that protein come from originally if I had the DNA? We have a chicken and egg problem here. Big time chicken egg. If the information to make the DNA, make the protein is in the DNA, how do I get that out to begin with? because we haven't had a chance to get to it, all this involves using proteins to even make all these structures. This is a picture of one of these nucleosomes, and it shows the DNA. You can see the helix. Can you see the helix, everyone? It wraps around those proteins, and they have a specific structure. Now, what's really interesting, I'm moving along quickly. What do you think holds the proteins together? It's the hydrogen bonding, and the dispersion forces that hold them together. Hold these molecules together, large proteins together, and what's even more amazing, if you look over here, you can see there's hydrogen bonding going on between the protein inside the nucleosome, all right, and the DNA. So the DNA, the hydrogen bonding and other forces that we talked about are responsible for holding it together. What I want to do, this is a picture of the same thing with water molecules. It turns out for DNA to be stored on these histones and to then work properly, it needs water in with this huge structure. How do you think the water is being held there? It's being held there by hydrogen bonding, the forces we were talking. It fits, they all fit exactly. All right, I'm going to try and move here quickly because I have the video does summarize this. And we're not going to look at the infrastructure of these molecules. We're just going to again talk review what's going on here. First, you have to do the transcription, which we talked about before. RNA polymerase does that transcription to make messenger RNA. Now, it's the messenger RNA that then goes on to the ribosome. That's a picture of the RNA polymerase. There are about 3 different types in a cell, and they range from 8 to 14 different proteins to make this structure. This is the RNA polymerase. This is the workhorse that's doing it. And what it's doing in here is it's spreading the DNA apart. It's making the RNA messenger RNA coming out here. That's transcription. It's just taking DNA going to RNA. There are locations right outside the nucleus. That's where these ribosomes are, and that's where we're going. The messenger RNA is going to these ribosomes, where we're finally going to do the translation. Now, what's happening here is the messenger RNA goes into the ribosome, all right, and out comes the protein. What's involved here is proteins, messenger RNA, which was resulted from the first step, and also this polymerase is also making some other goodies. It's making ribosomal RNA and also making transfer RNA, and these are all needed to do the final translation. This is just a picture of ribosomes. Let's just look over here. What's in our bodies, in all our cells. The ribosome is composed of 80 proteins. Not just 80 proteins. It's composed of 4 RNA molecules, long molecules. One of the shortest RNA molecules, these are all these bases, 1870 bases hooked together. And they form these structures together. So it's a mixture of protein and RNA, hence they call it a ribosome. If it's just protein, remember, it's an enzyme. It's an ACE-type molecule. Here is what a particular ribosomal RNA, this goes in again to make the ribosome. That lists all the different bases in order. It's 1,500 and some, 1,522 bases lined up. But because of hydrogen bonding, this is the structure you get. This is a secondary structure you get. And it lines up, you see, if you look closely, right in here, and we're getting very close to the end, hang with me, there's hydrogen bonding holding these together. All these hydrogen bonds are critical to the structure and the operation of the ribosome, which is going to do this translation. There's another piece that goes into the ribosome. This is the transfer RNA. I like to call it the translate RNA. You can see, here we have RNA. This is an RNA molecule with some special bases in there as well. There's the amino acid, and down here is the code for the amino acid. So if we could back it up, on the RNA, now unfortunately we're reading, you have to read right to left. You have to do a little Hebrew on me here, all right? In the RNA, it was TTC, and when it split apart, this was the coding DNA. When it split apart, this was the template, made a copy of AAG, and because we're talking ribosome here, this is UUC, otherwise it would have been TTC, right? And then, of course, then it matches up, again, by hydrogen bonding, holding them and matching them together. So you can see there's actually four times there's a hydrogen bonding sequence going on. And I've just shown them right there. And if you remembered, this is going in the other direction. This is the amino acid it was coded for. What's amazing, now the colors there are all the amino acids that these are coded for. And there's 20 amino acids, as I have over here. And what is really amazing about this, one important point I have to make, it turns out that this code is optimized. Meaning, even if I have one of these, I get a mutation. Even if I get a mutation, there's a very good chance I'm still going to get the same amino acid. All right? So it's very unique. Now, there's a guy named Herman Yaki. He said that the genetic code that we have is 10 to the 70 types of possible codes by the calculation. He says, but it's more probable that we have 10 to the 18. And if you read down the bottom, see that 10 to the 18? That's 1 with 18 zeros going that way. Simon Conway Morris, a noted evolutionist, he says this code, the genetic code, has eerie perfection, meaning it's built to prevent bad mutations where you get the wrong amino acid. This morning I said in hemoglobin, you get the change of one amino acid, sickle cell anemia. You can hardly read that one. This is Francis Crick. Biologists must be constantly keeping in mind that what they see was not designed, but rather evolved. It's a frozen accident, meaning we had to have this genetic code right from the beginning. You can't get away from it. The origin of life appears at the moment to be almost a miracle. And that was said back in 1981. Bill Gates, everybody hear Bill Gates? What he had to say about DNA. Human DNA is like a computer program, but far, far more advanced than any software we've ever created. That's what Bill Gates said. Now, he knows what he's talking about when it comes to programs, right? If something was created, somebody had to write the DNA program, right? You know where? I have a book up here, Programming a Life. It's a Christian brother. He has a PhD in chemistry and information science. If a computer processes information, where did the information come from? Our friend Richard Dawkins, Boy, that's hard to read. The machine code. Remember, this is a DNA protein machine he's talking about. The machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of computer engineering journals. He even acknowledges this looks like a computer. But it all happened by accident. Now, with the aid, and this is the second to last slide, we are going to review the DNA translation as it probably should have been done first. Let me see if I can bring this up. It's just 2 minutes. 3 minutes, we'll be done.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperone. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperone achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
As you can see, protein synthesis is an amazing process which occurs constantly in our bodies. Now, get this. Ribosomes are made from RNA and proteins. So the ribosomes are made of the very proteins they manufacture. In fact, it takes over 150 existing proteins to manufacture just one protein. So in essence, it takes a protein to make a protein. It's this chicken and egg scenario that continues to baffle science.
Final verse, or slide, I should say. And this is easy to remember because it's Psalm 139. First one is 13, right? It says, "For you formed my inward parts. You knitted me together in my mother's womb. I praise you, for I am fearfully and wonderfully made. Wonderful are your works. My soul knows it very well." It's interesting, knitted. The thread of life, that's DNA. Now, I apologize, I thought I could add that initial hydrogen bonding stuff, and that got me behind the 8 ball. And so we had to rush a little bit there with the other stuff. And I really appreciate you dear people hanging in with me on that. And I'll hang around for questions.
Let's give Dr. Rathman So I'm going to ask Joe, you want to go ahead and grab a couple of people? We'd like to take up a love offering to Dr. Rathman to help him defray his costs. And so we're going to go ahead and do that and ask them to come forward and pass those plates. And so if you would like to participate in helping Dr. Rathman this cost, please just go ahead and put something in the offering plate as these guys come around. Now, let me just kind of wrap up for you what I heard Dr. Rathman saying. The question he asked originally is a watch a good example of a cell? And I think based on what you have seen, the intricacies in a cell are far more complex than any watch that has ever been made. If it takes a watchmaker to make a watch, who must it take to make a cell and the components of a cell? And so I appreciate Dr. Rathman sharing that with us and showing us that point. I never imagined how complex cells were until I saw what some of, and this is just a little bit of the information he's been sharing with us about how complex cells are. And so this morning he shared that a single cell is as complex as a city like the city of Knoxville. And we have millions of those in our bodies, and they all work together. That's what speaks to us of a designer. I'm going to pray with you for just a second, and then we're going to take a short break, and Dr. Rathman will be in here for question and answers. We have a book table at the back if you want to check out some of the resources. Please do that. Thank you for being here with us this evening. Father, we thank you for the opportunity of seeing the complexity of the design you have placed in the universe. Lord, we pray that we can just take away with us how incredibly complex you have created ourselves and how it all works together. Lord, we thank you for that evidence and for somebody like Dr. Rathman who can come and share with us about your incredible work. Lord, we just ask your blessings on him as he completes his presentations the next couple of days. Lord, I pray that people will listen to what he has to say and that your evidence will be demonstrated. Thank you, Lord, we pray in Jesus' name. Amen. So if you want to take a quick break, run to the restroom or whatever, and if you have questions, come back in here and Dr. Rathman will be available to answer questions for you.
Thank you, ladies. You're praying for me, I know that. Go. Thank you. I appreciate it. Yeah, well.
To you can ask the question, Dr. Rathman will answer it. So, questions? Questions? Okay, right over here. Nancy.
I have to go there, being somewhat challenged.
Okay.
I gotta talk in this?
Yeah, you do.
Okay.
Because I have hearing difficulties.
Oh, got it. Okay. So I've read somewhere that one DNA molecule contains 1,000 books of information,
Oh,
and I've heard 6,000 to 8,000 books of
Yeah.
information. So which one is closer? And either way, it's amazing.
you can figure it out for yourself. DNA has 3.2 billion pairs. And if you count each one, you divide that in 3 and count each one as a letter. So take 1 billion letters, and how many letters can you put on a page? I don't know. Well, figure that out. Once you figure that out, you can figure out how much you put in a book. Now, a billion is a lot. You think million? Well, a billion. On the web, you can see pictures of $1 million and then $1 billion. You go, whoa, because it's 1,000 times, it's 1,000 millions. It's 1,000 millions. Oof. That's why when you start putting those zeros on there quickly, it gets out of hand. Remember I said there quickly, Yaki said, well, there are, his calculations, there's 10 to the 70 possible genetic codes, but would you please give me 10 to the 18? As I mentioned, the gal standing there, that probability, its number of possibilities is also probability. That's equivalent to winning 3 lotteries in a row.
Isn't anything over 10 to the 18?
That's not when you have to have sequence, but just 3 lotteries. Huh?
Anything over 10 to the 18 considered theoretically impossible?
That's another game they do. Everybody has their own numbers. Bill Dembski, he says 10 to the 150. Some people say 10 to the, some people also say 10 to the 18. Richard Dawkins says 10 to the 18. Yeah. If you give me the DNA protein machine. Well, wait a minute. How did you get that? This is by natural selection. Well, what does that mean? It doesn't mean anything. Yeah.
Well, I agree, I understand, but I was just trying to put it in something my brain could comprehend.
What are you, again?
When you use a figure with
Yeah.
a chain behind it, all these zeros, 18 and over is theoretically impossible.
It's possible. It's more possible than 10 to the 50, right?
Yeah.
A lot of statisticians say 10 to the 50, that's it.
Okay.
When people come up with these numbers, I don't like to play the numbers. I tell them, you give me the number you're happy with. I'll take, the one, we didn't talk about it, but one protein, cytochrome, is only 110 amino acids. And it's actually 100, to make that protein, you have a possibility of 10 to the 143 possible arrangements. But he says, I want 10 to the 75. I'll take it. And it's amazing, they always try to get below 10 to the 80. You know why? Well, actually I should say 10 to the 82. That's the number of protons and neutrons in the universe. So anything over, I think anything over 10 to the 80, you should forget it. Think about trying to make, you make one, but you have to make millions of that same one. We're only talking about the probability to make that one protein. You need many of them to make a cell, because all these have a lot of the same protein in there. It's even magnified even more. You see what I'm saying?
Yes. Do any of the atheists tell us which came first, the chicken or the egg? Would they have any hypothesis for it at all?
They can't, because everything in the cell is needed. Everything. All the components in the cell, not only amino acids. The big experiment in 1952, when Urey Miller experiment, they made amino acids 2%. That was their best yield. And the problem is, it made a mixture of L and D. And they still teach this in high schools. You have to have only the L amino acid in order to have life. You get one D in with your amino acids in your protein, all bets are off. You have disease. That's why your body turns over your proteins. Every 7 years, you're a new person. Because all your proteins in your body have turned over. Because proteins have a way for each carbon, where the R group is, each carbon has a way to flip to the D. And that's where a lot of diseases come from. So your body has to turn them over. So even if you got the amino acids, all the L amino acids, and you put them all together, you only have a certain amount of time to make life, because some of those amino acids are going to go to the D version, and that's lethal. I already hit that hemoglobin. In one of those fragments, 114 amino acids in one of those fragments, one of those amino acids changes sickle cell anemia.
Okay, let's go on to, Daryl had another question over here. Dr. Rathman, let's go over this way.
You don't have to come up here. Real quick, and you know my background, you were with the NSA, am I correct? You're telling us that the body is, for lack of a better term, is coded, right? Are you familiar with the Enigma machine? Do you remember that from your work?
The Enigma machine?
Enigma. It was a code machine, okay.
You're talking about the World War II?
Yes, yeah.
Oh, yeah.
And how many...
We were spies over there.
So, how does the body, looking at, how many, I don't know how to formulate this, forgive my ignorance, but the different parameters that machine could have, different codes, the body is coded. Is that what you're telling us?
Oh, yeah. That table I showed you,
yes.
You can also put it into a circular form. And it looks just like the days when, I want to have a message. I put a G in, I put a T in, I put a C in, up pops the amino acid.
Okay.
It's like a decoder if you put it in a circle. I had that in there this afternoon and took it out. Unfortunately, I should have taken out a lot of the elementary stuff, but anyway, that was my fault.
But for us, there are just so many codes. You mentioned this. The number's phenomenal.
Oh, yeah. It's 10 to the 70, according to Herbert.
Okay, another question?
Herbert Yaki. Oh, by the way, the guy Herbert Yaki, who's quoted his numbers all the time. Everybody's familiar with Yaki? You saw him up there. Remember, he gave the number of possible codes, genetic codes, 10 to the 70. He's an atheist. He's trying to tell, the reason he comes up with all these numbers, he's trying to tell the government we should stop spending money on SETI. Because they're not finding anything. We need to spend that money here on other research. His numbers are telling us what the probability is to have life somewhere else out there. So he comes up with these huge numbers, millions, all right, that the probability of life is out there. What he doesn't realize, he's actually calculating the probability for us to even be here. If it's us plus somebody else, it's that probability times the same probability if it's carbon-based. He didn't even... But he's trying to get everybody's... The SETI project, search for extraterrestrial intelligence.
Search for extraterrestrial intelligence.
Yeah.
Okay.
E.T. Yeah.
Dr. Rathman?
That's so bad. Maybe we should have just done Q&A, huh?
Hello. The way I understand it, there is intelligent design, and then there's chance.
Yeah.
And pretty much by now, everyone's given up on the idea of chance. I mean, think
That's right. They're trying to get away from that natural selection. Exactly.
What about this third thing I'm starting to hear about? Physical necessity. What do you think about that as a...
I'm unfamiliar with that. But you do bring to mind something that I'd like to expound and would help you maybe a little bit. Richard Dawkins, he realizes the problem of coming up with a DNA protein machine. He'd like to have it. You give me that, and I can make all the complexity in the world. All right? He says that. And I forgot where I was going, but where was I going?
About Richard Dawkins needing a protein machine.
Yeah. It's been a long day. This is how he solves it. His chemical selection, single selection of one amino acid at a time to get the protein we want. He has a way to do it. Something that's 100 amino acids long, this is the way you do it. If you look in the book, it's called Methinks It's Like a Weasel. He writes that out, but in different letters. All right? Now, he knows the target. Methinks it is a weasel. It's like a weasel. And so what he does is, you can imagine whatever the number of characters there are. I have a slide for it, but we took that out too. All right, so it's maybe 20 characters and spaces. And so what you do is you spin the dice, and they all change. But what he does with his algorithm, all the ones that fit, methinks is a weasel, he keeps those. He selects that. It's like one of these kind of drawings.
You can't do that.
You can't. Then he spins it again. He's getting closer. Hey, I got 10. I achieved 10 to the whatever. What was it? 10 to the 40. No, 10 to the 140 power in 43 spins of the dice. But he's not blind at all. He knows where he's going. It's in this book. I can't believe it. I fell off my chair twice. Which means I was back up on my chair. So, and he calls that selective. No, he didn't call it selective. He calls it cumulative selection. Of course it's cumulative. He adds it up as he goes.
Who's doing the selecting? Yeah.
He's the blind watchman.
Intelligent design.
Exactly. You got it. He's doing the selection. What's so blind about that? Are you kidding me?
Next question.
By the way, as far as I know, he hasn't given anybody the program to do that. But anybody can figure out how to write it.
I just have a quick question or an observation, really. So what is happening, like when Dr. Ross was here and explained how we are at such a good time to see the beginning of the universe and see the things that have happened, and with all the advancements in technology and everything that's happening, where are we in this scientific community? When are they going to break and say yes? When are they going to acknowledge the presence of God as
It's
the
will.
designer?
It's all about the will. They're already trying to get away from natural selection. To me, that's sort of like, well, it just happens as natural selection. Next question. Oh, that just happens by natural selection. Next question. That's their God of the gaps. They just let that. And then when they finally, like Dr. Ross talks about physicists, when they finally figure something how it works, oh, we don't need God. Hey, we got the Higgs boson. We don't need God. If you didn't have God, you wouldn't have the Higgs boson to study. Plus, you wouldn't have the brains to figure it out. And it turns out the Higgs boson is just as designed as it is at that level on up to the universe.
Right.
I don't know when they're going to do it. I don't expect them to do it because it's all about will. And the smarter people are, the more excuses they can come up with.
Okay, next question. Dr. Rathman, back here at the back.
So we have all these processes going on, right? Within a cell or within DNA and all these structures, right? What is the motivating force for these actions? I mean, we have neutrals, positive, negatives, forces, but what motivates these things to do their actions.
All you have to do is, for the cell, all you have to do is feed amino acids in the form of protein, fats, fatty acids, and carbohydrates, and it keeps going.
And they just keep doing the work.
It just goes.
They just do the work.
Right. That's why I was saying it's so much different than a clock, which has to be wind. It's so much different than a mouse trap. Yeah, a mouse trap's great. It's irreducible parts, but you still got to set the trap. And somebody's got to come up with the cheese. There's a lot more going on. And Michael stops at a very simple spot, which is good enough. But you just have to give it the foodstuffs. It'll make its own ATP. That's not STP, but ATP. All right? And that drives it. It just goes. And it replicates. And as long as there's not any mutations going on, with time you're going to have mutations. What's really cool about DNA, and in the transcription step, and in the making of the RNA, all right, and in the translation step, there are editing enzymes in there. They see mistakes, they fix it, and actually, what happens too, it takes. It turns out that when it finally makes a protein, that yield is only 80%. So what happens is they say, well, why would God do that? It's only 80%. Goes through all that and only make 80%. What happens, some of those, the 20%, they get cut up into amino acids in small pieces. They get sent to the cell membrane. And there is the immune system. They're checking, hey, this comes from us, this comes from us. So a virus comes in, it tries to take over and make its own proteins. It sees proteins very early. It says, shut it down. So it's built in for our immune response. Turns out, when something is optimized, some things in the part is not all that optimized. There may be other reasons, but for the overall workings of the machine to keep on trucking, everything is optimized. That's what makes it even more fantastic. The information is optimized. It runs like a clock. It runs like a computer. It just goes.
Wouldn't it be wonderful if we had self-repairing computers?
Yeah. Someday we will be self-repairing.
We would love to have one of those. All right, next question.
You've done an excellent job of proving what the Bible says, that a man does not believe in God is a fool.
Oh, yeah.
Yeah. And
sometimes
I marvel at how God gave David this inspiration to write that psalm that you put up.
Yeah.
And he didn't know anything about DNA or embryology or all these other things beyond the cellular level. But I have a very simple question. You mentioned free radicals. When I eat my blackberries, what happens to make me healthier and purge my body of free radicals?
Yeah, well, there's what they call antioxidants in there, and they scarf those suckers right up. It's the radicals that go after the DNA. Okay. And of course, if it's going to go after the DNA, that's where all the information is. Remember, there's two strands. One strand is the coding part, the other part is the template. We didn't really have a chance, and that's my fault, to talk about it.
Biochemistry.
It's
Memorized the Krebs cycle so many times.
Yeah, many times, yeah. I get $1.5 for that. Antioxidants, that's what they do is they, as you know already probably, that they scavenge up those radicals. It's amazing too, a lot of it comes from oxygen. Did you ever think about this? The most corrosive compound in the universe is oxygen, O2. It's two oxygen atoms together. All right? And if you had oxygen and you're trying to do a pond, make proteins, it's going to be oxidizing as fast as you do things. So how can we have a system that doesn't work in the presence of oxygen, but it uses oxygen? How could you develop a system that uses oxygen, yet tears it up? It has to all start together. You have to have the oxygen, and you have to have the organism to handle the oxygen. Did I lose me?
I light you to some other place
Yeah.
because of something you said.
Yeah.
Okay.
Well, you know, it's just amazing to me. Oxygen is the most corrosive, yet we need it. So how did it form in the first place? How did life form in the first place?
Rust tears apart steel.
Pardon?
Rust tears apart
Oh, yeah, sure, yeah.
Let's take two more questions, and then we'll try to finish up, and then if you want to talk to Dr. Rathman privately, you can go ahead and do that. So,
And again,
any other
I'm
questions?
sorry, I had 15 minutes ago, I felt like it was time to do a commercial, and I thought it was going as fast as I could. I thought I wanted to put the water in there. Maybe you got the message just from that. Water is highly designed. The way it's in, you saw in the central zone, it's needed every spot where it is. People have found enzymes that work fantastic. You take one molecule that's in that little cleft where the active site is, where it does its work, you take that one water molecule out of there, it doesn't work.
Okay, Dr. Rathman?
Yeah, I'm sorry, I was digressing.
Yeah, I don't know, this may be a theological question.
I'm into it.
Okay.
He's here now.
Okay. Is it that God created the body to function on its own?
Sure.
Or is he constantly supporting it and allowing it? Is God making our bodies work? Does that make sense? Is God supporting all the functions that our body, like all
You think
this,
he's actively
this
involved?
complicated
Are you asking, is he actively involved
Yes.
now?
In the body?
When it comes to healing, he can intervene, but he's not active now. He created so it would run on its own. And it runs, what's amazing too, as you probably learned from Dr. Ross, we need entropy to get fuel. Entropy is to get fuel. And yet it's entropy that is a result, causes mutations. So it's a catch-22. We need entropy to get fuel to live, but it's still the entropy going to a greater state of disorder. We need that to live. And entropy also causes mutations. Communication between us, you know, you do the little thing around, you tell a story around a circle, by the time it gets to you, you don't even recognize it. There's entropy in communication. The same thing happens there. You have information, sooner or later there's going to be a slip up. What happens, you can have a flat molecule stick in between two of the latter parts of the DNA molecule. You might get a misreading. And just changing one of those characters can give you a different codon, which gives you a different amino acid. Trouble city. But remember, the genetic code is designed, if you get mutations, it's designed that, it's not that bad a deal. It didn't work that bad. It's still going to work. But we're only here to last so long. Believe it or not.
One more question before we conclude this evening.
Did I?
Anybody else?
That wasn't very theological.
Okay.
When you're looking at stuff in the electron microscope
Oh,
on
yeah.
a molecular level, how do they separate a molecule or DNA strand to even look at it without the background being so, do they fill it, is there gas around it? What's around it so they don't...
Well, it's all done in a vacuum. They have to stretch it out. How they do it, I have no idea. But the electrons, when they're fired at the target, because there are electron clouds around the molecule, it deflects them, and that's how comes it disperses off of there, and then develops your picture because of this changing in this. It's just like taking a photograph, but with electrons rather than light. That's maybe why I should have started. I've never had an electron microscope. I wish I would have, but
Can't buy one of them at Walmart, unfortunately.
it's the X-ray studies that are really incredible. We only just showed you the part of the DNA going around the protein there, and what's so amazing, that is so fine-tuned. Remember, this amino acid inside on a protein just happened to have the right R group to reach up there and hydrogen bond with the DNA to hold it in place. If that isn't fine-tuning, I have no idea what else is. And that's why you can see if you had a different amino acid there where that one was, you're in trouble. You're already in trouble.
Let's give Dr. Rathman a hand. Thank you for being here this evening. Thank you very much. If you want to just if you have some questions you want to privately ask him, you can check out the book table.
I have some calendars back there, pocket calendars. There are no batteries in them, so you don't have to worry about running down. But I have my email address in there. It's called his-T-Links. All right? I used to call myself T-Links because T, Terry, Links, L-I, and N-K-S, I was always on the links. Never mind. Okay.
Thank you for coming tonight. Thank you for being here.
I appreciate
Thank you to
you all
Dr.
so much.
Rathman for being here as well.
Yes, ma'am.